100, chapter 12 and beyond. We're going over Mendel's experiments and heredity and some general genetics. Okay, so this is going a little bit beyond chapter 12 in your book, more like a little bit of chapters 12 through almost 17, but this is a topic that's really hard to fit into one lecture. It's a topic you could spend the whole year on, which I know I've said about every topic we've covered, and it's true about all of them, but genetics is even more crazy, in my opinion. But we won't get too far into DNA and things, but I do want to go over a little bit of heredity, how you inherit certain traits, what that means, and how it looks different for different people, different sexes, etc. So let's start with the basics. We have this guy, Gregor Mendel. I don't really care that you know too much about him, but basically he's kind of like the father of genetics, right? So he's kind of the first one, and he started messing with these pea plants. And he chose pea plants because they're simple. They have flower color, seed shape, seed color. And he considered these traits true breeding, meaning that basically you would see it in the next generation of offspring, right? So it was easy to manipulate matings. He had each pea was a new individual. They had short generation times. It was cheap. So it's an easy way to kind of study heredity and get some of these basic concepts down. Okay, so you will get a lot of vocabulary and notation in this lecture, so just keep that in mind. These might be good slides to refer back to. But whenever you see P, that is the parent generation, not just of true breeding plants, but as we start to go into the Ps, you're going to start seeing these associated with Mendel's work. So P is the parent generation. F1 is the first filial generation, so that's basically just the parent generation breeding and producing the first offspring. F2 is two offspring from F1 crossing and having children. This is F2, and then F3, 4, 5, those are succeeding generations. Siblings would be considered brothers and sisters. I think we know that. Cross sibs would be mating between brothers and sisters. A back cross is mating an offspring. Self cross is a plant with itself. And then true breeding is when parents have the same traits, always producing offspring having the same traits. So these last few here, I'm not super concerned with you knowing. And true breeding is more like associating this with your book. We don't really refer to true breeding very often when talking about genetics and heredity, but basically it's thinking that as parents you will have the exact same traits that you pass down to your kids. So we're going to look at some formal crosses for one trait. So say we have two pea plant parents up here, and then these are the offspring that they're going to produce, and then we're going to cross these offspring to produce F2, right? So we have the parent generation, F1 generation, F2 generation, and we could keep going on and on and on. So to do a cross, we call this a formal monohybrid cross, just meaning don't get too caught up in this language. Basically, we're just going to cross these two parents and form hybrids, okay? So we have a purple pea plant with a white pea plant, and then we cross them and we get these purple flowers down here. But they must be hybrid, right? Because we had a white plant mixing with a purple plant. But there's no blending that we see here. So now if we cross these two and we get our F2 generation, we see we've got three purple plants and one white. So we have both of them showing up here. So how does that happen? And that's when Mendel really started to think, okay, how are these traits being passed down? Why does one show more than the other, etc., etc.? So in this experiment, he crossed plants that were true breeding for violet and white. That's the P generation, right? And then F1 had all violet. F2, we had that 3-to-1 ratio. So basically, this told Mendel that we don't just blend inheritance, right? Like if you mate with another male or female and you produce offspring, it's not just a literal blend. So if you have black hair and brown hair, it's not just going to be like a brown color. it's that inheritance appear to be particulate. So basically the idea that we have dominant and recessive traits. I'm sure you've heard of these. So dominant means that that version of the trait always shows through and can then mask other versions of the trait, which would be recessive. So in this example, you can see the recessive trait is masked by the dominant. So the white is masked by the purple in the F1 generation, but in F2, those recessive versions now can actually mate with each other and produce a all recessive plant where you actually see that shine through over the dominant version. Okay, so we have our parent here, and you can see in this case we have both a dominant and a recessive allele. So again, more vocab, and we'll get to a vocab slide here in just a second, but Mendel created a few different laws. One is that he had a law of segregation. So when an organism produces gametes, remember gametes are sex cells. So gametes are sex cells. A male would have the sperm, a female would have eggs in humans and lots of other animals as well. So for male sperm, female eggs, the two alleles for any given trait separate so that each gamete receives only one allele. So you can see here we've got our purple parent and our purple parent, and they have a phenotype, which means what's showing. And keep in mind, phenotype isn't always, it's often called like the observed trait. It doesn't always show. A phenotype could also be like that you are susceptible to cancer, right? That's not something that we can see. We can maybe pick it out with certain tests, but we do often refer to phenotype as like the observable trait. So our phenotype here is a purple plant and a purple plant. The genotype is this right here, the capital R, lowercase r. When it is capitalized, that indicates that it is dominant. When it is lowercase, that indicates that it is recessive. So each one of these, each letter, is what we call an allele. And because, if you remember, we've talked about this a little bit, gametes, our sex cells, only have one allele each. So basically, as a parent, I have this genotype. I have capital R, lowercase r, but my eggs can only have either a capital R or a lowercase r. So basically, that's what I can pass down to my kids. I can either give them a capital R or a lowercase r, and I'm going to mate with someone up here who has the same genotype, coincidentally, and his sperm is only going to be able to give out a capital R or a lowercase r. And then we're going to do this thing called a Punnett square, which is what this is. And we're going to cross these to see what the potential is for our offspring. So we have capital R, we're going to bring that down. We're going to bring over our capital R here. So we have a capital R, capital R. So we have a purple flower. And we have a capital R over here, bring down a lowercase r. So here we're still masking that recessive allele, right? And we still have a purple flower, and we see the same thing down here, but now we bring down our little r and bring over our little r here. So now we have two recessive alleles, which gives us a white flower. So that was Mendel's, not really his first law, but that's one of his laws, is segregation, is that each parent can only give out one of these alleles to their kids, right? Which makes sense because as, let's say, kids of our parents, we have these two alleles. So that means we got one from mom, one from dad. He also had another law that said, it was the law of independent assortment, and it said that any two alleles for any given trait will segregate independently from any other alleles passed on to gametes. So basically, I know this gets a little bit complicated, this Punnett square here, so don't mind that there's more, but basically now we're looking at like two different, let's call them genes for the sake of this class. Two different genes here. We have whether you're smooth or wrinkled, and whether you're yellow or green, right? So we have a smooth yellow parent and a smooth yellow parent, and they're going to give off one allele from each gene. So they can either give out a capital R or a lowercase r, and they have to give out either a big or a uppercase Y or a lowercase y. So they do have to give out one of each of these, but they're independent of each other. So just because you give out a capital R doesn't mean that you give out a capital Y. You could give out a capital R and a lowercase y, or you could give out a capital R and a capital Y, or a lowercase r and a big Y, right? So these are independent of each other. It doesn't matter which one you pass down. Okay, so he had this physical basis of these particles. So what would the particles turn out to be? We just saw. So here's again some more useful modern terms that we use. So a trait is a characteristic like flower color or eye color, right? Again, a lot of times it's observable, but not necessarily. Um, and then the phenotype is the characteristic version of a trait that we actually see. Again, we don't always physically see it with our eyes, but a lot of times we do. So a phenotype would be like it's a purple flower, or you have blue eyes, right? And you're mating with someone who has green eyes. Allele is, like I said, it's that capital R or that lowercase r, right? It's a specific version of a hereditary particle. So a hereditary particle, basically, from what Mendel was saying, today we would call that a gene. Okay, so we have different alleles in genes, and this can get really complicated, but for the sake of our class, we'll consider one gene has two alleles. Okay. Dominant, again, this is alleles that mask others, usually has a capital letter, so like capital P for purple flower. So if there's a capital, it's always going to win out over the lowercase or the recessive. So the recessive is the one that's masked by others. So in order for a recessive to show through, it has to have two recessives, which we would call homo, right? Homo meaning same, hetero meaning different. So if you are a homozygote, you would have both alleles of the same trait. So if you have two capital P's or two lowercase p's, you are homozygous. Keep in mind that homozygous can be either dominant or recessive. So both of these are what we would call a homozygote or homozygous, but we would say this is homozygous dominant, this is homozygous recessive. On the other hand, a heterozygote means that the alleles of the same trait differ. So we've got one capital P, one lowercase p. Okay, these are heterozygotes. This is describing their genotype. So we already went over the phenotype, which is what we see for the most part. The genotype is the combination of their alleles, basically, that are carried by an individual that causes a phenotype. So genotype is the letters. That's telling you what alleles that person has in their genes. And that is what's causing the phenotype that we see. So let's say I have blue eyes. I have two little b's, right? Lowercase b. That would be my genotype, and it's causing a phenotype of blue eyes. All right, and up last we have zygote. Zygote means the first diploid cell produced by fertilization. We've gone over that a little bit, but basically you have haploid cells, your gametes, your sex cells. So one sperm and one egg, when they get together and fertilize, they produce something called a zygote, which will later turn into an embryo, a fetus, and then a child, and then an adult. Okay, just a few more terms. Gene, again, this is in the DNA strand that encodes information causing a trait, right? This is what Mendel referred to as the hereditary particles. So this is the one that has two alleles in it, which again is just different versions of a gene. Locus is a place of interest on a chromosome, usually a gene. Okay, DNA is our double-stranded helix, right, that you've seen before, I'm sure, but we don't talk too much about it in class because we just don't have time. And we won't really go over these three. They're not super important for you right now. Okay, so doing some more crosses, if we cross our purple flower, that is a purple homozygote, meaning purple, dominant purple, with a homozygous recessive white flower, so little p, then we're going to get our F1 generation of heterozygotes. So I'm going to go ahead here and draw a Punnett square. So I always put dad on top. Let's say that dad in this case is our big P's over here, our purple homozygote. And then we have mom over here, our little white flower. I'm going to put little lines underneath because otherwise it's impossible to tell what the, if it's big or little, basically uppercase or lowercase. So again, I'm just going to carry these down, right? So my big P is going to come down. I always put the uppercase first just because that helps with it, but basically I'm just drawing them all out. So I have 100% heterozygous kids that are possible from this cross, right? And then if I take those kids and I cross those kids and I have big P, little p, big P, little p, and again, we'll do this. Then I have where I get my 3 3s. So I have my big P, big P, big P, little p, right? Big P, little p, and then I have little p, little p. Okay, so this is my 3 to 1. So I have my purple, my purple, my purple, but I have one homozygous dominant, two heterozygous, and then I have one homozygous recessive, which ends up being my white flower over here. I know this is a lot if you don't remember Punnett squares, so we will get some practice in our class activity that you guys are doing, or I shouldn't say class activity, but assignment. Okay, so with Mendel's first law, hereditary is particulate, meaning that we have like dominant and recessive, basically, and that they recombine randomly during fertilization to produce offspring, and that we can only pass down one of these alleles, right? So then to test these hypotheses, that is when you do the crosses like this and get to the Punnett squares. So Punnett square is basically just a way of figuring out what offspring you could potentially have with these parents. So again, we kind of did this already, but our parental gametes we put here. Again, I usually put dad on top and mom over here on the side. Doesn't really matter, but you will see that that seems to be kind of the standard. And then you always put the uppercase first because the uppercase is just easier to indicate first. It's like a universal standard so that you know if you do have a dominant allele in there. Okay, so oftentimes to verify these results, we will do test crosses, and that's what Mendel did as well. Determine whether an organism expresses a dominant trait, whether he's homozygous or a heterozygote. And so Mendel did that, played around with these, and basically got all of these different things that we all know today. So our Punnett squares that we do can basically predict what our cross outcomes will be. So we can see, okay, there's a 3 to 1 chance that we're going to have yellow offspring and a 1/4 chance that we'll have green, right? And that's why the Punnett squares are useful. And you can do the same thing technically if you know the genes that you possess and your partner, and you could technically go through and say, okay, our offspring would have this, this, and this chance of having these genes, right? Or these phenotypes, let's say. Okay, I'm going to skip over this. The dihybrid cross just means now that we have two different genes that we're crossing. So just kind of showing you that it exists, but I think it's too complicated for how little time we have. So I won't have you guys do this part, but just know the monohybrid, which is just one gene times one gene. And if you're interested in this, you can go ahead and take a look at this in some of the other chapters as well. And we've gone over the law of independent segregation, which is where this really kind of comes into play, where it doesn't matter which allele you're passing on, it's not going to be tied to the other. Okay. Okay, so when none of Mendel's laws apply, like the independent assortment, what we call that is complex inheritance. So Mendel is simple inheritance. Now we're getting into complex inheritance, which for the most part is sex-linked usually, or sex determination, whether we're male or female, sex-linked inheritance, and some of these other things as well that we'll briefly touch on: incomplete dominance, codominance, continuous variation, multifactorial inheritance, and non-disjunction. Okay, so getting into sex inheritance, one of the, or sex inherited traits, basically, one of the ways that we do this, we can use Punnett squares still, but we also use something called pedigrees. And pedigrees basically combine probability rules with this thing called a pedigree to show males, which are the squares, females, which are the circles, and when they mate, they're drawn here with a line. So we have a male meeting with a female going to produce these three offspring. And then, like, let's say these offspring find someone special that they want to mate with. Like right here, we found another female we want to mate with. And then we have two offspring produced from that. So you can see males, females, if they are affected, which means that they possess this trait that we're doing a pedigree for. Like, let's say this was for baldness, and dad was bald, mom was not, and then they had offspring, and they had one non-bald child, male, one female bald child, and one unknown, unaffected or not bald child. Okay. So here's an example for alkaptonuria, which is black urine disease. It's a really rare inherited disorder that basically prevents the body from fully breaking down two different protein building blocks or amino acids, and it results in a bunch of acid basically being built up in the body. But you can see the pedigree here. We have an affected male who has this disease, and basically usually when we're doing the pedigree, honestly, most of these are recessive. So it means that he would have two lowercase alleles, right? Not always, but for the most part, most of these like disorders and diseases that we see being represented in pedigrees are usually recessive linked. So you can see here it is recessive. We have a little a, and then we have a mom who he's mated with, who in this case, we're not quite sure what the genotype is, but they had one son who was affected and mated with a unaffected female who was heterozygous or a carrier for this trait. So keep in mind, females can be carriers. So you can see she's got one dominant and one recessive allele. Because she has one recessive, she can pass that on, but males cannot be a carrier for the trait. They either have it or they don't, and I'll explain that here in just a second. All right, so what determines sex determination? This is obviously a really complex topic, and it's not to be taken lightly or that one thing matters more than the other. So going into this, I want you to know that we are strictly talking from the biological sense. So we have sex hormones that are produced by gonads, which is ovaries in females, testes in males. And then within the testes, or within males, I should say, we have androgens like testosterone being produced. Within females, we also have androgens. We have testosterone and estrogen. Males also have testosterone and estrogen, but they vary in the amounts, right? So males produce higher levels of testosterone, females produce higher levels of estrogen. So what actually determines what shapes the development of our sexual anatomy? Well, that depends on the set of chromosomes that the fetus receives from the parent. So again, we have gametes or sex cells coming from males and females. So females have an XX, they have an X chromosome and an X chromosome. Males, you have an X and a Y. Okay, so mom can give up either an X or another X in her eggs, right? Because we can only give up one, right? So this is basically like her allele, you could think of it as, but it is chromosome. Males in sperm, you can give up either an X or a Y. So then depending on how these mix will determine whether you have a male or a female. So a female would have an X from mom and an X from dad. A male would have an X from mom and a Y from dad. So boy or girl, the Y chromosome is actually the one who decides, right? So dad is the one who decides every time, which is why we always say whether you're going to have a male or female offspring, a kid, that's going to depend on the father. So what determines the actual biological sex? So on the Y chromosome, there is a gene called SRY, or we often refer to it as the sorry gene, and that is what signals testes to develop. Testes then produce testosterone, which is in turn kind of creating the male sexual anatomy. Having said this, there are XY chromosome disorders that can be caused or affected by the loss or damage of the sex chromosome. It could be duplication, it could be deletion, etc., of those chromosomes. So then the phenotype does not match the chromosomal sex. So when we're talking about biological sex, in this case, really what we're referring to is chromosomal sex. There are genetic, hormonal, anatomical, and behavioral aspects to sex, and we've heard, it seems like, especially lately, lots of talk about this, right? What qualifies a male or female, and how you identify. And this is strictly, again, in the biological or chromosomal aspect. But there is research, and we'll get into it in just a second, or I'll kind of touch on it, that sometimes things happen, right? And that's kind of the driver, the explanation for how people are feeling. So females, again, you have an XX, which means that you have ovaries and your genitalia is female. Males, XY, testes, male. They do have pseudohermaphroditism, which is XX still, but you have ovaries and male genitalia, and you are otherwise infertile. So hermaphrodites are usually infertile. I shouldn't say usually, they are infertile. And then male pseudohermaphroditism is where you have testes, but you have female or ambiguous genitalia, and again are infertile. Intersex, which is a true hermaphrodite that has both. Hermaphrodite, depending on who you ask, is sometimes sort of offensive. So they've since kind of started calling it intersex instead, which is when you do have either XX or XY, but you are born with both ovaries and testes, and you have both male or female, male and female genitalia, or they're ambiguous. So infertile, again, historically called true hermaphrodites. And there is interesting research and things happening with this. Hermaphrodites born at birth are very, very rare, but there have been cases where the parents decide, okay, we're going to make them a male or we're going to make them a female. Some parents just let them be hermaphrodites, and then they can decide later in life when it kind of, it's a little bit more of their personality is showing through and different developmental stages, and you can kind of understand. And there have been unfortunate cases where doctors decide and say, okay, we're not going to tell them they're a hermaphrodite, and we're just going to make them a male or a female. And there's an interesting, I use a textbook in one of my other courses, an interesting story in there about a girl who, or sorry, now I can't remember, but she was born a hermaphrodite, and they decided that she would be female, and so they let her grow up as a female, and from a very early age, she did not identify with that. She always thought that she was more of a male, and at one point she was getting really sad and talking to her parents, and I think very early on, like as early as 8, was saying, I want to be a boy, I want to dress like a boy, all this stuff. And then as a teenager it kind of came to a head, and that's when her parents told her that she was actually born hermaphrodite. She had both parts, and they decided to make her female, which I think is often what they do. They, I think, tend to go towards the female side. So she then sort of, I don't know if she biologically transitioned or just started identifying as a male, but there are things like that that go into some of these behavioral and hormonal aspects of sex that are something that your body has been fighting against its whole life, right? And so those are really interesting cases. And like I said, there's a lot more that goes into sex than just the biological component of it. Talking about lots of different things and how we consider gender identities, but just the different chromosomal identities that we can have. We also have something called triple X syndrome, which is where you actually have 3X, and you are a female, you do have ovaries, and you are fertile, but you tend to be taller than average and tend to have learning disabilities as well. There's Klinefelter syndrome, XXY, 47XYY syndrome, which again is a male. but more learning and emotional disabilities. Turner syndrome, just one X, is the female. Infertile, broad chest, webbed neck. These are all very rare, but these do happen. We have seen them in medical literature. Okay, so when we're talking about sex-linked inheritance, that means that it's a gene located on either of the sex chromosomes, X or Y. So daughters and sons don't share the same probability of inheriting it. In addition to being called sex-linked inheritance, this is often called X-linked inheritance. And it's because it's referring to X-linked traits, which is a phenotype that is determined by an allele on the X chromosome. So for recessive X-linked traits, you can have a normal copy on one X chromosome that masks a recessive disease allele on the other. That's why we say that females can be carriers, right? They can have, like, let's say for baldness, they can have a normal X-linked trait for that makes them normal, and then the baldness X-linked trait that does not show through because it's recessive, but they can pass it on to their son or daughter. Another example is Duchenne muscular dystrophy, or DMD. So you can see here sex-linked inheritance with the mom who has a DMD allele. And then dad who's unaffected. So he has a normal X and a Y. Mom has a normal X and an affected X. So she is a carrier for this. She can pass it on to her son or daughter, but she's not going to show this trait. Dad is unaffected. He's just going to be able to pass down an X or Y, right? So the odds of them having an affected child are 50% because we have mom's affected X over here and one daughter and one son. So the odds of an affected child are 50%, right? We have 2 out of 4. What about an affected daughter? Again, also 50%, right? Or sorry, affected daughter. Oops. Okay, let's back up. The chances of having an affected child are 1 in 4, right? Sorry, there's only one that's affected. The chances of having an unaffected child are 3 in 4. The chances of having a daughter who's affected is 0%, right? We have a 100% chance of having an unaffected daughter because we're always going to have this dominant allele thanks to dad. So even if the daughter gets one from mom, at most she can just be a carrier. Okay. So the only one that we can really pass this X down to is the son. And because males only have one single X chromosome, they're going to show the effects of any recessive alleles located on the X chromosome. This is something we especially see with like color blindness because color blindness is X-linked. We see a lot more males that are colorblind because we have a lot of females that have one normal gene or one normal allele and one affected, which means that they're normal, they're showing the dominant allele, but they're passing down the affected one to their son. You can see the other example here where dad is affected and mom is not. And again, now our chances of being affected are 0%, basically. If mom did have an affected allele here, we would increase our chances, obviously, but because dad's passing down the Y, he can't possibly pass that to his son. But he can make his daughter a carrier. Okay, so mom is passing one of her X chromosomes. Father is only passing his X chromosome to his daughter and his Y chromosomes to the sons. Boys only have one X chromosome, and so they have a higher probability of inheriting X-linked diseases than girls. So we can use a pedigree to follow the inheritance patterns, again, which is just a visual representation of the occurrence of phenotypes across generations. So we can see who's affected and who's not affected, males and females, and in the affected here in the blue. So again, symbols for the pedigrees, we've gone over to some extent. Male is a square, female is a circle. If they're affected, usually they're colored in, and if they are not affected, then they are blank. And the little diamond here is gender unknown, and then a line through is deceased. These ones are less common to see in pedigrees because sometimes, it really doesn't matter. Okay. So let's talk about the Y chromosome really quick because that is sort of interesting. The Y chromosome contains very few genes and is really, really small. So it's really often used to study ancestry and also identify paternity, right? Who's the dad? Sons inherit the Y chromosome from their fathers, and we can compare the DNA sequences to fathers and grandfathers alike because the Y chromosome. So let's back up a second here. The X chromosome does go through something called crossing over. Crossing over basically just means that the X chromosome is changing. So the X chromosome from mom to daughter or dad to daughter is not going to be the same X chromosome that you inherited from mom or dad. There's going to be different genes on it that have been crossed over. The Y chromosome, on the other hand, does not go through this, and it's basically unchanged. So the Y chromosome is the same for dad, grandpa, great-grandpa, great-great-grandpa, all the way up. Because of that, if any of our dads mess up or if we need a paternity, we can actually compare something called short tandem repeats or STRs on the Y chromosome. I won't get into what this is. If you're interested in it, you can definitely look it up. But basically, we can look at the Y chromosome of any males in the family because they're unchanged. So an interesting story about this is the Thomas Jefferson pedigree. You can see Sally Hemings had 7 children, and the DNA matched through the inheritance line of her youngest child. They did that, and they matched, and this is a slave, by the way. One of her slaves had 7 children, and it was always hypothesized that maybe Thomas Jefferson fathered them, or at least some of them. And so they did an STR analysis of the Y chromosome, and the DNA profile of a male descendant of Jefferson matched that of the kids from this slave, like descended down. So because the Y chromosome does not change, it is very likely, or we should say at this point known, that one of the Jeffersons did father those children. Was it Thomas? Maybe. Was it his brother? Possibly, right? The more likely scenario is obviously Thomas Jefferson did it. So that's kind of an interesting take on the Y chromosome. And then getting into some of the other things that we can have, and that's under complex inheritance, is something like incomplete dominance. Incomplete dominance is basically when things mix. So if we have a red flower with a white flower, and instead of getting our dominant would be normally red, right? And white would be recessive. But instead, when they mix, now we get a true mix, which is pink. Okay, so the pink would be the heterozygote. So we would have one red because we have one homozygous dominant, one white because we have one homozygous recessive, and then 2 pink because we have 2 heterozygotes. Also, we can have multiple alleles for a single trait showing an order of dominance. So we won't get too far into this, but some traits do have multiple alleles that go into it, or homeotic mutants, which can affect development and potentially many characters. So this is single genes that affect only one trait. Was from Mendel, right? But now we're saying, okay, there's multiple genes that can affect one trait. For instance, drug resistance in the malaria parasite, right? We can have, that is not controlled by just one gene, that's actually multiple genes. Sorry, I'm going to skip this slide, it shouldn't be right there. But again, going through inheritance of sex-linked traits, I think some of these slides got messed up, so I'm sorry. But we can go through this again as an unaffected father and then a carrier mom. Remember, dad can't be a carrier. Dad either has it, he's affected, or he's not affected because he's only got one X, so he can't hide anything. Mom can hide stuff, right? She can look normal on the inside, but still have half of her that she can pass on that could affect her future children. There are also lethal alleles, right? Some alleles that are so non-functional that they'll kill the individual inheriting it. They can be dominant or recessive. Some examples are cats here. There are some examples in humans as well, and that's usually something you go through in pregnancy and not necessarily at a later point in life. This is the crossing over I was talking about with the X chromosome. Sorry, I think my slides got mixed up somehow, but that's okay. You don't need to know too much about this. Just know that the Y chromosome does not go through this, which is why the Y chromosome basically looks the same. But we can see the X chromosome here. It's basically switching, like if we have one from mom and one from dad, and then they're just going to switch some of the genes on here so that they are different in the offspring. I'm not going to talk about this too much. Again, this is something that you can go into if you're interested, the epistasis, where genes are independent, but are not independent, but they interact to produce different phenotypes. This is getting into the dihybrid and trihybrid crosses and stuff where we're talking about multiple genes. So it is really interesting, but we just don't have the time for it in this class. So back to incomplete dominance. Again, this is kind of like if we cross red and white and we get pink, or if we have two different versions of a gene that specify hair texture, but we actually have three phenotypes, right? So we could have a curly-haired kid, a straight-haired kid, or a wavy-haired kid. So straight would be the recessive, curly would be the dominant, but if these two mate, then we would get like a wavy-haired kid as the incomplete dominance. So again, this is like if we have the curly and the straight haired, and we have the wavy hair as the heterozygote. In addition to incomplete dominance, we also have something called codominance. Codominance, there are different genes that are codominant, but one of the most popular ones that we hear about is for blood. And transfused blood has to be compatible in lots of different ways that are determined by genetics to actually donate blood or to receive blood from someone. And so if you know the blood types, you know that we have A, B, and O, and then we have positive or negative, which we'll talk about here in a second. But codominance means that both the maternal and paternal alleles contribute equally and separately to the phenotype. So there are three different blood group alleles, A, B, and O, and the genotypes are as follows. So we have OO. O is actually recessive. So here we have a homozygous recessive. Here we have AO, and since O is recessive, it's just A. BO, which would just be B. But here, A and B are actually codominant. So our blood type would actually be AB. AA, that's homozygous dominant, and BB is homozygous dominant as well. But when A and B are together, it would be blood type AB versus O, A, or B. So there's four different blood types, O, A, B, and AB. And then we also have, this is just explaining that further. Blood type alleles, A and B, again, codominant. O is recessive to both A and B. So you can see if you have type A markers, you are either a genotype of AA or AO, depending on your parents. Type B, you're either BB or BO. Type A and B, you would be type AB. And if you have no blood markers, you would be OO. So you can see who you can donate to and who you can receive from. OO is actually the universal donor. You can see that they can donate to each different blood type, but they are only able to accept from type O's as well. So they're the universal donor. The universal receiver is type AB. So if you have AB, you're actually really lucky because it means you can take blood from anybody. The other thing that goes into blood type is something called rhesus factor or RH factor. This is where the positive or negative comes from. And basically, whether you have a presence or absence of these proteins determines whether you are positive or negative. If the donor and recipient, again, are not compatible in their blood type and rhesus factor, then they can have a life-threatening immune reaction to donated blood. So that you wouldn't be able to do it. You do have to match the blood type, and then you also have to match the rhesus factor as well. Okay, guys, I know that was a lot, and it's just scratching the surface on inheritance, but we are all done with inheritance, so I hope you enjoyed that and learned a little bit about traits you've inherited from your family and traits that you could be potentially passing on.